Plasmalogen, a type of phospholipid making up cell membranes with accounting for up to 20% of the total mass of phospholipids in our body, was first discovered in lipids isolated from starfish in 1909. Later, in 1924, Thogan and Voigt named the compound plasmalogen to describe its producing plasma aldehydes after acid treatment. As a unique kind of membrane glycerophospholipid, plasmalogen took nearly 40 years to accurately establish its structure. Thus, research on its action in the human body continues to this day. Plasmalogen, being produced in the liver and transported to other organs through blood vessels, is particularly abundant in heart and brain cells, playing various roles. Although oxygen entering the human body through breathing acts basically beneficial to the body, free oxygen radicals are produced to this process. Excessive free oxygen radicals promote to the oxidation of cells, that is, aging. Inhibiting such oxidation of cells is called antioxidant. Plasmalogen plays an important role to endogenous antioxidants that protect other phospholipids, lipids, and lipoprotein particles from oxidative stress. Moreover, it regulates various intracellular responses, including activation of platelets, excretion of cholesterol, and promotion of cell membrane fusion. According to a 1999 study by an American Association of Neuropathologists, decrease the structural modification of phosphatidylethanolamine plasmalogen in the brain in the Alzheimer's disease. Total phospholipids in the frontal cortex and hippocampus in the brain of a patient with Alzheimer's disease decreased approximately by 20% DNA-based. This change was caused by a decrease in the phospholipids such as phosphatidylethanolamine and phosphatidylcholine. The low content of phosphatidylethanolamine was due to the decrease in plasmalogen. Through these research results, it was found that the modification and oxidation of phospholipids were associated with Alzheimer's disease, which was published in the Journal of Neuropathology and Experimental Neurology. Since then, research on the relationship between brain disease and plasmalogen has been conducted steadily. A 2001 study suggested that plasmalogen deficiency plays an important role in the pathogenesis of Alzheimer's, and a 2007 joint study in the United States, Canada, and Japan demonstrated that plasmalogen levels were reduced in the blood of Alzheimer's patients. Takahiko Fujino, an emeritus professor at Kyushu University in Japan, noted the change in the human body when taking plasmalogen while researching treatment methods for disease caused by the brain fatigue, such as diabetes, depression, and dementia. According to his 2012 thesis, dietary plasmalogen increases erythrocyte membrane plasmalogen in rats, which was published in Lipids in Health and Disease. He explained, based on previous studies, resulting in a decrease in plasmalogen in metabolic and inflammatory diseases, such as diabetes, heart disease, cancer, respiratory disease, and Alzheimer's disease. He conducted an experiment to measure change in plasma levels by feeding diabetic mice to a normal mice's food. As a result of the experiment, he found that plasma cholesterol decreased in both diabetic and normal rats, and the level of plasmalogen in the red blood cell membranes were relatively increased. In addition, the same change occurred even when plasmalogen was fed continuously for 9 weeks, with no side effects observed. This suggests that plasmalogen intake helps reduce plasma cholesterol as well as raises plasma level in the red blood cells with no abnormalities or side effects from such direct intake. He also studied the decrease in plasmalogens in the brain tissue and blood of Alzheimer's patients. First, after confirming the effects of plasmalogen on the animal Alzheimer's model, he conducted a randomized placebo-controlled trial in patients with mild cognitive impairment and mild Alzheimer's. As a result, he found that 1 milligram of plasmalogen intake was effective in Alzheimer's patients. Furthermore, he evaluated the MCI patients previously reported based on the mini mental state examination, Japanese. It's a Japanese simplified mental state test to evaluate cognitive function and dementia in the elderly. In this study, he divided the MCI patients previously reported in the placebo group and the plasmalogen intake group to evaluate the change in MMSE-J score over 24 weeks. As a result, the total MMSE-J score of the plasmalogen intake group was significantly improved statistically. In particular, the cognitive ability of place, space, and direction was improved compared to the placebo group. In this study, it was demonstrated that oral administration of 1 milligram plasmalogen improves cognitive function of MCI patients. Moreover, as a result of comparison by immunostaining in an experiment to examine the effect of cranial nerve cells conducted in 14 aging-promoting mice, it was found that cranial nerve cells were emerging in the plasmalogen-treated group. That is, plasmalogen intake helps cranial nerve cells grow and increase. This was important in the development of cranial nerve cells in the dentate gyrus, which is an area of hippocampus being responsible for memory. As a result of the above experiment, it's expected that the cranial nerve cell generation of the dentate gyrus will have the effect of treating memory impairment, as well as improving learning memory and ability. As such, research on plasmalogen is steadily progressing. Through the result of a number of previous studies, we know that plasmalogen is not only effective in improving cognitive impairment such as Alzheimer's, but also because it's extracted from edible biological tissues, it's being reconsidered as a highly stable brain nutrient with almost no side effects.